We can take up a couple of questions.
Yeah, if you allow me, I can make because there were comments ask questions in the chat about the the case study. So I would like to mention something, how the case study was designed and the difficulties we have to implement it. So the case study have different purposes. Today you have seen how the case study of growing fish of the same origin in two different locations in East Mediterranean and West Mediterranean. They have been used for the purposes you have seen today. The other objective of the case study was to perform at, let's say, pilot scale, something research that was done previously at the lab by our colleagues from the Work Package 2. So they were testing some feeding and feeds that that they suggest in their experiments. And one of the main conclusions we got is that at pilot scale we got very similar results to the laboratory laboratory results. So for us that was a good conclusion to see that there are say the feeding experiments that the researchers conduct at the laboratory, they can be directly applied at the farm levels because we didn't find the differences. Just about the case study, one of the main difficulties we have, it was to let's say to replicate or to have two sites, let's say with standard conditions. So it was not easy to find two farms that could devote 16 diameters cage. So they had to devote a farm for this purpose. They had to follow different procedures in the with the feed in the feeding regimes. The feeding regime was the same, although at the end we found that there were slight differences and and fish were fed a little bit more in Greece, although the feeding regime was similar. And as regards environmental parameters, you have seen that it was taking temperature, oxygen, probably there were other parameters that now we can consider that could be important to monitor, as water current, for example, turbidity or light. So those could be recommendations for future studies. For those of you that didn't attend last week webinar, our colleague from Wageningen, Arjan Pastral, made a very interesting presentation about the influence of water current and growth. So the presentations are already from last week webinar are already available at the Medate web page. And the presentations that you have seen today, they have been recorded and they will be available on the Medate web page. Okay.
Bernardo, maybe we can take out some of the questions that have been to the whole group.
Please.
Shall I do it? Okay, yeah.
Yes, please.
Yeah, you're taking this? Okay, Anna. Go to the questions and answers as well, please, because there is one for Beatriz and one for Beatriz and Ross.
Yeah. I will just take them randomly. So one is from Francesca Pfeffer. Is there, to Ross and Beatrice, is there any use for millions of snips filtered out and not used for the 60K in population genetics? For example, association with some signature of domestication. Maybe you could answer to everybody.
Sorry, Anna, I didn't understand the question.
No, well, you can read it, but I can.
Where is it? Just a second.
Yeah, in the questions and answers.
Q&A. Is there any use for the millions of SNPs that have been filtered out and not used for the array for population genetics? Maybe Ross.
Ross,
Me to
yes.
start,
You can
and then
start, yeah.
I'll pass to Beatrice afterwards. So yes, indeed, there's a database of millions of SNPs, and also all of the whole genome sequence data. Bear in mind that the data we generated are from pools of DNA from populations. So they're a pooled seq data set, and they're all available linked from the publication that I showed in the presentation. So people can access those data. But to answer more directly, as well as what I said there, that I think, yes, there is potential to look at things like signatures of domestication, and I believe there is some ongoing work on the data and using some of the same populations that we worked on. But I'll allow Beatrice to take over in case she has more to say on that.
No, basically what you just said, yes, probably the more you have, the more accurate you could have these studies done. Yes, I agree. Yeah. For things like, looking at selection signatures and all of that. I mean, I'm not very sure if probably with the information that we have at the moment, we could do, we could get some accurate results, but probably with more data, we will have a better accuracy for that.
Yes, there's been some studies before which have used pooled seq data for looking at signatures of domestication. So technically, I think it's definitely possible. But obviously, the one limiting factor may be the ability to categorize populations into domesticated versus wild in a clear way. That may be a challenge for some of the populations, I think, because of in some cases, the early stage of domestication.
Okay.
Yeah.
Thank you. John, would you like to say anything more on the questions you got on the two standardization of the two environments? You've got several questions on that.
Yeah, so the explanation that Bernardo already gave is that we standardize the husbandry conditions as much as possible with the same, giving the same feed, having the same size cage. It's interesting that you also mentioned the swimming results that Ayapalstra presented, because I was at both of the sites for the sampling, and there is quite a difference in the current at the Spanish site compared to the Greek site. And yeah, we haven't measured it, but yeah, you just notice it when you're there.
Yeah, just for information, regardless, the Spanish site, it was in more open water conditions, where the Greek site was most protected.
Yes, yes. Then the data collection, of course, there was a question still from Dinos that I didn't answer yet, because it was a bit difficult to type the whole story, which was specifically for fillet weight. How standardized was that? And so we didn't measure it with the machine. We had to go to 2 different countries. So in Greece, the company has a packing place nearby. So the same person from the packing that does filleting, the commercial filleting, as a job. So very much a professional, did the filleting of all the fish. And but then we realized when we got to Spain that we needed to do it ourselves, and it's quite a intricate job to get right. So we put the same person on each day, but we see quite an effect of the person that is doing it on the fillet weight. Of course, in the model we can account for that, and with that we still see that we get a relatively high heritability of close to 0.5, 50% of the variation that we see, we can still explain with the genetics, the genetic relationships between the fish. So yes, especially fillet weight was difficult to get consistency in the measurements. Of course, harvest weight is easy to get consistent measurements, but yeah, some of the others it's more difficult. But given the heritabilities that we got, any noise that you introduce with the measurements, it reduces the heritability. So given the heritabilities that we got, we probably did okay.
As Bernardo said, if somebody is interested in the case study, you can go to Alicia's step presentation from last week in work package 2, and then you will find the design and the parameters taken and details of what you are asking about feeding rates, temperatures, and so forth regarding just the case study, which would help to understand the background, the context where John worked for the genetics. There was some differences in the appearance of the fish, for instance, at the end between both sizes, and not very significant, but there was some small differences in there. And then I would recommend you to go to that presentation for the more fine-tuning issues. Anna, you want to carry on?
Yeah, we got another question. Why is a common and sectorial improvement program in the Mediterranean so complicated compared to the salmon industry? I can just make a couple of comments on that. I hope the other speakers can say more. I think it has to do maybe with the matureness of the industry and how the ownership of these companies is going. Remember, here in Norway, we have a couple of very specialized breeding programs. They provide material to the whole sector. There are also a couple of integrated companies that also have big breeding programs now. And that is the structure that is mainly used in the sea bream and sea bass industries. So it means that you come from a production company that will have to actually introduce a whole new methodology in the premises that they're already doing. But I think already from the beginning of this project and until where we are now, has happened a lot in these industries, at least the industries I know of in Greece and Spain and so on.
Yeah,
but of course not in all the countries, and they are at the different stages.
Yeah, and if I can add something, and then that is related with Beatrice's presentation, with what you are mentioning, and with Beatrice, the structure of the sector is quite different here in the Mediterranean. There are more countries producing on-growing fish, more countries with hatcheries. So meaning that the sector is more dispersed. But one of the things that we were looking at the beginning in the study of the diversity of the populations is to see whether farm populations were different from wild from farm. And for me, it was very interesting to see that we could see such a clear differences because that was not clear at all. During many years in the Mediterranean, hatcheries have relied on wild fields broodstock, and there are still companies working on that way. The fact that we see such a big difference with these SNPs may point out that even though we have not done a great work on breeding, maybe we have done important work on domestications. This question, there is relating with this, a question in the chat from Victoria to Beatriz about these issues, asking whether she would like to know if you could go in depth about the potential effects of escapees in wild populations. And which do you think is the main potential risk at genetic level for both species?
Yes, I think I answered to her already, but I don't think we have much information on the amount of escapees that there are, right? I might be wrong, but I don't think there is much information on that. So, but it's clear that this is, as you just said, that the structure of wild and farm populations is different. So, if there are lots of escapees from farms, that might create a problem because of it can change the structure of the wild populations. But first, I think that we will also need to know how much, how big is this problem of escapes from farms.
Yeah,
Maybe somebody knows from the panel.
and let me say something, and then I move to Dinos. At the beginning, when we were taking the samples, at least in Medate, we were asking the farmers about the origin of the fish, and that was a tricky question, and we didn't have clear answers. And we saw in some cases that the hatcheries farmers have moved broodstock and fish from one place to another, meaning that well, that was a common practice. Maybe Dinos can comment on that.
Yeah, I think it's very difficult for some of the farms to know where the fish that they have coming from, because some of the small ones that do not have a breeding selection program, they have never kept track of where they're getting their fish. They kept getting the best fish, the best performing fish from their cages, and using them as broodstocks, or one year they get from one source, another year they get from another. It's only now, at least in Greece now, I mean the last let's say 5-6 years, that the big companies have started with their breeding selection programs. We have now, I think, 1, 2, 3, 4, 5 breeding selection programs in Greece. So we have more than Norway. And those companies, they have obviously tried to get breeders from known sources, and they know where they're coming from. But the majority of the smaller hatcheries, it's difficult to know where they get them from.
We got one question for Siri. If you can repeat the sample number for the gilt-head sea bream to estimate heritability, and if you have tried to run a bivariate model for the traits.
Yes. For the number of samples for body weight and visceral weight, we had 967 fish, but for the fatty acid traits, we had 232 fish. And yes, I used a bivariate model in ASRML to estimate the genetic correlations, but not the heritabilities.
Then we got a question from Magdi. He wants to know optimal conversion ratio and what the growth rate are. And I think we are here working with industry data. So I think I wanted to answer this. We cannot give out information on this. So, but maybe Magda, you could join our stakeholder group. There we are going to meet industry partners. Maybe you can get direct information from them, but it's difficult for us to give out commercial data information.
Yeah, if I can add, well, in the pilot case study, if you go and look at the presentation, we have the conversion rate, and our sister project, Perform Fish, is working on that. So hopefully in the near future, they are going to present results about so technical performance from commercial farms. So.
Yes. I wonder if we have more questions here. No, I don't think so.
Well, here there is a question,
Agree.
maybe for you. I don't know. Is asking from Nagaraga. Is asking how many generations taking place in this selective breeding program.
Very worked on here. John, maybe you can answer for the Galaxy D.
So for the sea bream that were, well, it's also a difficult one to answer because of sources of fish and so on, and the exchange that that Dinos in the past that Dinos mentioned. But there is 2 to 3 generations of managed breeding program, but yeah, not starting from wild fish. So that's the difficulty in answering this question.
I think it's the same for the sea bass.
Okay.
Yes.
I don't know if.
I think.
Yes. Okay. If there are no more questions.
I have a question regarding
Okay.
the wild fish. How sure we are that this wild fish that were sampled are wild? For example, for Cyprus, sea bream and sea bass is not a very common fish in the wild, not as much as it has been in the Western Mediterranean. So I really I wonder how wild those fish are. How sure we are that they're wild. And okay, they were collected from the wild. That's one story. But how close to farms were? Could we tell anything about their size? Maybe they were escapees, which is very, very common. What do we know about the wild, the origin of these wild fish?
I will say one thing, when we started to do the analysis, I mean, for instance, thanks to Bernardo and other people, we found out a lot, no, not a lot, but some errors in the identification of the population. So I will say that it's interesting because in the end, everything was more or less clear, okay? So I presented here some populations, wild populations that they were completely different to like the Cyprus, like you say. And then when we asked the people responsible for these populations, they say, okay, actually that is a tricky one because the sampling was done in 4 different locations and so on. So, I mean, what I was trying to say is that by looking at the results, these genetic results, we can see all of them basically make sense, and before some of them didn't make sense. So when we check for these populations that for which we were getting strange results, and then some of them were the problems were solved, and some others no, because for instance for this Cyprus population they told us no, in fact they were sampled from different places. So the same with the effective population size. We will expect that if all the wild population have basically the same structure, actually when we analyze this wild population separated from farm populations, the optimal K for sea bream was 1. So basically it's telling us that basically it's one big population. So if one of them have a very low effective population size, we will think that there is something strange there, right? So what I'm trying to say that the results from this genetic analysis tell us a lot about if the sampling was done properly or not in some way. I don't know if that makes sense what I'm just saying, okay? If you find a population with a completely different pattern or a different effective size to the rest, probably it's because there have been some problems with the sampling. Yeah, so I agree. But the rest, for sea bass, for instance, it was clear that they were all behaving in the same way.
Okay.
Okay.
Have we answered all the questions?
It seems so. So Well, before we finish, we would like just to make a very short poll, just to know about the use of the simultaneous interpretation. I don't know if my colleague Diego has launched that poll. Just to know if the participants are using the language interpretation that we are providing, and if they are, which language they have used. So this is just for organizational purposes. If we finish, just to remind that the recordings of this webinar are going to be available at Medate web page. And I invite you, well, you had followed these presentations, but you have heard as there are related work that has been presented in a previous work package webinar. So I invite you to visit our web page and follow results from other work packages. I think that they have nothing more to say. That's just to thank all the panelists that have joined us today for the excellent work they have done. I don't know, Dinos, Dolores, or Anna, you would like to add something before we?
No, I would like to thank everybody for listening. Hope you found it interesting with genetics work. At least we in the panel are very interested in this and hope to get more breeding into our industry or in the industry in the world.
I would like to add there, I think maybe the people attending have seen my my question and John's answer, which I think it's maybe the most important thing that we saw today, after, apart from also the population genetics, that even with G and to E interactions, even with the breeding selection not being optimized for different sites, it does make sense to use genetically selected fish as opposed to, you know, unselected fish from your locality. And I think this is very, very encouraging, and I think this should be broadcasted to the industry, because maybe by doing this, we will develop an impetus for one or two companies that will focus on breeding selection of sea bream and sea bass, and then they will be selling maybe parents or maybe eggs, or I don't know what, but we'll get away from this 5 breeding selection programs in Greece, and then we'll get into 1 or 2 major selection programs for this. I think this is at least to me, maybe I'm a little bit naive in this, but at least to me this shows that you can have a breeding selection program in Greece or in Italy or in Spain, and you do it under your conditions, and the fish will perform better anywhere. Maybe in some places better than others, but there'll be better fish to use than unselected fish that you choose by yourself. So I think this is something that for me, and correct me if I'm wrong, I think this should be broadcasted and should be emphasized, because maybe this will give some strength to developing such a company that we all been wondering, as Cristobal said in his message, why isn't such a company available for this?
Yeah, and just something to add on this, in Medate, in Work Package 6, where we are working on economics, our colleagues from Cantabria are working in the development of a tool in order to try to assess the economic impact of different parameters, like how, for example, the performance on feeding, on growth, other parameters can affect economically a farm. So for those are tools that are under development, and farmers, I think they should look at those tools, because a difference of 5, 10, 20, 30% in the growth is a lot of money. And this can be estimated.
Thank you. Yeah, thank you, Dinos, for asking that question, because if I send a message that using local unimproved strains is better than improved strains from somewhere else, then I missed my point very much. No,
you didn't.
But thanks for asking
exactly
that question.
The right. You pointed exactly the right thing. No, I think.
And on top of what you're saying, so if there is this development, which you've seen in other industries, that there are small breeding programs more local, and they agglomerate into bigger breeding programs. What I want to add is that if you know about these slightly different needs for different production systems, one breeding program can incorporate that knowledge and make sure that you use data from more than one site. So there is also ways to, at least in part, mitigate in your breeding program this diversity in production systems that you're serving, which is for a local breeding program difficult to do. But if you're a larger player and selling to different parts of the industry, then yeah, that's definitely the way breeding programs go.
Yeah. Well, it came to my mind something that is important related with this work of the differentiation between wild fish and farm fish, is that not now, but something that 10, 15 years ago, they were samples of restocking exercises in the Mediterranean for sea bream and sea bass. Looking at the results we have got today, one recommendation would be if an administration would like to do that restocking of fish, sea bream or sea bass, that it is better to use for those purposes wild fish and not farm fish, in order to not to genetically impact in the populations. This is something that, for example, 10, 15 years ago, 20 was not taken into account. And with the knowledge that has been shown today, that will be an important recommendation too. Not to use farm fish for restocking purposes for this species.
I would like to say that I am really pleased from the beginning of the project, talking about developing tools for genetics, which they are most needed, as we saw from Anna's introduction presentation. I think we can be very pleased of the work done. The tools are there, as Dino says, it should be broadcasted to the sector and for their use. And also the implementation of those tools is proving very successful. So I would like to congratulate you all in the work done. I think we can be, as a project, feel very, very proud of what we are providing to the industry and to all stakeholders, and also emphasize that it's in open. So everybody can use it. So it's been paid with public money and it's for everybody to use. So I think we can all be extremely happy about that. I am personally, and I thank you all for your work.
Good. Okay. Thank you very much. We are on time, which is.
Actually, we got one more question.
One more question. Okay.
I think it's for Beatrice and Ross, maybe. Suppose improved and unimproved fish is mixed in any population. How can we observe any kit is there? Well, maybe, yeah.
I don't think you can. I mean, in the admixture pictures, you could see, if you look at the detail, in one particular population, you might have an individual that it has completely different genetic composition to the rest. So maybe you can see it like that. We saw it in some of the populations, that the proportions from the different ancestral population were different for maybe one individual or two individuals. So might be that is the reason. Yeah, so you can see things like that in the admixture plots. Yes.
Mm-hmm. Yes. Yeah, there was an answer here from Victoria. There are several tools for detecting if an individual is wild or farm based on morphology, scales, ortholiths, etc. These variables may be used together with genetic analysis. Yes.
Mm-hmm.
That's a good clarification, Victoria.
Mm-hmm.
Okay.
Okay, I think that we can close the webinar. Thank you all panelists, all participants for following us today. For the participants, just let me remind you that this webinar is going to be uploaded at the Medate webpage, where you can find a lot of information from already published the deliverables or publications. So you are interested in the topics that we have presented today or other related topics, please go to the webpage of the project.